Good morning, Lana. Good morning, Rose.
Good morning.
You still have allergies?
I think I'm getting sick actually.
Oh dear. Hopefully just a sniffle.
Oh,
Hello, Simon.
Hey, Professor, how we doing?
Doing well. Last class before the last exam.
Ooh, I know, yeah.
It's exciting. Okay, so three of you, you have me all to yourselves. Any questions you want to ask?
I have one question.
Uh-huh.
It's gonna be on chapter 18, I believe.
All
So I have
right,
it
give me
written down
just a
right
wee
here.
bit. Oh, let's see. PowerPoint 18.
Yeah.
Oh, that would be the one that isn't actually available in my list. Ba, 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 bum. All right, far away.
All right, so it's right at the very end. I kind of get it, but what's like the difference between the insulators and the silencers?
Oh, okay. So um, insulators separate one gene's regulatory region from another's,
Okay,
so that um, the effect of any enhancers or even silencers binding to one regulatory region does not affect that of another genes, right? And the big deal about that is that DNA can bend,
gotcha.
right? In fact, it has to bend for um, transcription to be activated, right? So if you look at this slide, that's a very stylistic but accurate representation of how expression is initiated, right, with these enhancers. And so if you can imagine, there's let's say there's another promoter kind of further to the right, maybe this piece of DNA could bend a little bit further, and then that enhancer could um, activate another gene's expression, right? Or it could bend, the other way and do the same for a gene going in the other direction. And so essentially, insulators are kind of almost like silos. They're little walled gardens, right? And so they say, okay, this bit between here and here, anything binding to that DNA is going to affect only the expression of the gene within these two walls, essentially, right? And anything that binds here is not going to be able to kind of jump over and affect this gene over on the other side, because that DNA is prevented from bending. It's made stiffer, essentially. Right, so that's an insulator. A silencer is much simpler in many ways. It's simply a protein that binds to any one of these regions, either an enhancer binding site or transcription factor bind site, whatever, right? And prevents them from binding. And so if you want to reduce gene expression or even prevent it entirely, at least at a single gene level, then presence of those silencers will stop either the assembly of this transcription apparatus or the binding of enhancers, let's say, that help actually activate transcription. So really insulators are more about, kind of changing the structure, not the structure of DNA, but its ability to change shape, right? So that what happens in Vegas stays in Vegas kind of thing, right? Whereas silencers are like another one of those dials and switches. Just the same as transcription factors and enhancers can, turn on or increase transcription, silencers can turn it down again. And so the output in terms of transcription is a balance between silencers and activators and transcription factors. And actually transcription factors can be both activators and silencers. It's really just that they bind DNA and affect transcription.
Okay, gotcha.
So really, one of the things I wanted to get across in this particular PowerPoint and lecture or lectures is the astonishing degree of complexity that there is in control of gene expression in eukaryotes. I mean, in bacteria, it seems pretty complex, particularly if you haven't seen it before. But eukaryotes is just truly astonishing how finely controlled, like the complexity of control of transcription or gene expression in general. And that's really in many ways what gives us our complexity. Not just as like I'm a special individual kind of thing, but in terms of biology, that's by and large the reason why, with the same number of genes as a nematode, right? I don't know if you can kind of see it, like a worm that's about that long, right? We have the same number of genes as they do, and yet we are considerably more complex than worms are, thankfully. It'd be really weird teaching a lecture as a worm. And the reason for that is the extra control over transcription and gene expression we have over worms. And the reason why worms are more complex than bacteria is because of the extra control that they have in terms of transcription over bacteria. Makes you do more with less, more with the same amount, essentially. Did that help?
Definitely, yeah, thank you.
Good.
Cool. And then just one more question. Is the test process going to be the same as our last exam? Or
Yes.
Okay.
It will be online. I'll send an announcement out with actual concrete details, but essentially it's going to be two smaller exams, one of which is just on the material we've covered since exam 3, and the other one which is kind of over the whole semester, but a fairly shallow level by and large. It's really more to assess your progress over the semester. And so don't panic and think, oh crap, I've got to learn everything going back to the very beginning of the semester. To be honest, if you don't know that stuff by now, then extra study, I'm not sure, will make a huge difference. It's really to assess, as we've gone through the semester, how much of this stuff have you understood?
Gotcha.
How much has your knowledge increased?
Same exam or two different or
There will be two separate exams. I haven't quite figured out how to wrangle that yet. Yeah.
I'll just wait for your announcement. No problem.
Yeah. Well, before I always used to do it as 2 separate paper exams with 2 scantrons, and so then I can just grade each scantron separately and be able to analyze the data separately. I don't want it all mashed in together, because then for every student I've got to pick out every single. I have to calculate the grade by hand essentially, and that's just no fun whatsoever.
Yeah. Sure.
Yeah, so it will be two multiple choice online exams. It will be available throughout the whole day on Thursday. So if you have scheduling conflicts, and I'll put this into the announcement as well, you'll have the opportunity to take it whenever you want during that day.
Oh, okay. And that's gonna make it a lot better. Yeah.
Yeah, and almost certainly you'll be able to. Well, you would do the exams separately, right? You wouldn't have them both open at the same time, because that'd be nuts. But you don't have to take them linearly or in a particular order.
Gotcha. Okay.
There'll just be 2 separate links, and you start one, and you have a certain amount of time, which will be, half of the class period essentially, and then you'll have the other one.
Okay. Alrighty.
Yeah, but I'll send around an announcement. It's on my list of things to actually do today.
Definitely. All right. Sounds good.
Yeah.
Alrighty. Thank you, sir.
No worries. Alana, Rose, you must have questions.
Right now I just have a really general question. Did you have a meeting like this on Thursday? Because I couldn't find it anywhere.
Yeah, I did. Let's see. Oh, you now get to see. There we go. Oops. What date was? Yeah, 23rd. It's there in the channel. Come on, YouTube.
Okay.
This one here. I could play it for you, but it'd be kind of weird to hear myself.
Oh yeah, that's fine. I was having problems finding it yesterday. I don't know why.
No, it should be. I mean, I don't know how you view this because, you know, this is kind of my channel, so to speak. But yeah, it should everything should be in here in the
Alrighty.
genetics playlist.
I'll just look for it one more time.
Yeah, if you have any, I didn't label it wrong, did I? Hang on, I wonder if I gave it the wrong name. That might have been the reason why. Let's see. No, 234. That one, that's weird. It's not popping up in the.
See, that's where I was looking at it, and it wasn't popping up because the link that you gave us sends us to like the page that you were just at before you went back to this one.
Oh boy, now I've got to actually. Let's see, did that just not go into. There should be a simple fix in theory. Just give me a second. Easier to do. Oh God, no, I don't want to do that. Let's go back here. All right, so you know what would be easier is shit bits. Oh, that's right. There we go. Probably some of my astrobiology students like, what the hell have we got genetics in this?
Oh, there it goes.
Stuck in the wrong playlist. Hopefully, let's go back. Oh no. Oh, there we go. Save. That's what I want. Now. I really need a bigger screen. There you go. There it is down at the bottom there.
Alrighty, sounds good.
Yeah, thanks for that, Alana. I never realized.
Yeah, no problem.
Okay, let's get rid of that. Oh, bollocks. Here we go. All right, what's next?
I had a question about knockout and knockdown genes. What's the difference other than one doesn't destroy the gene, I guess, in a sense, completely?
That's exactly the difference.
Okay.
Let's see, where was that? Transgenics, I believe. Yeah, so a knockout is anything where the sequence of the DNA is permanently changed. And so typically that results in the, lack of function. Or it doesn't have to, right? So you can get all kinds of different mutations that do all kinds of different things genetically. And loss of function is just one of those changes that can happen. But it's a permanent change, a permanent heritable change. So if I got one of my worms, which is a knockout, essentially it's a mutant strain, right? So it has a sequence change relative to the wild type, which is kind of normal, whatever that is. And that sequence change results in a change in function of a gene, right? You know, and that obviously we'd know what that gene is. So every single individual in that population has that same change. Every cell within the worm has that same change, right? So it is kind of, it's everywhere, right? 100%. Unless you do like funky stuff with the genetics where you have a heterozygous individual for whatever reason. Some of the lethal genes, for example, you can't have them as homozygous recessive because otherwise the individual dies. You have to maintain them as heterozygotes, which is a royal pain in the ass. But the issue there is that because every individual has that and every cell has it, it's very powerful, right? Because it means that the effect is as strong as it can get because it's everywhere, right? And it's as penetrant as it can get for that particular change. You know, every individual is going to display that phenotype because they have that change. Now, the issue is that that kind of broad brush kind of ties your hands in several ways. One way is that if this gene is involved in, let's say, an early developmental process, right? No, and let's say it's involved in multiple processes. There you go. That'll probably be an easier thing to get. And one of those is an early developmental process, right? So without that gene function, that organism won't develop to the point where you can see the effect of the later phenotype. Does that make sense?
Mm-hmm.
Right. So it's almost like kind of self epistasis in a sense, right? You know, the earlier phenotype affects the expression of the later phenotype because the worm never develops to that point, or the structure doesn't develop, or whatever. Another issue is, let's say you had a gene which affects a lot of different processes, and you only want to look at the effect of that particular change on, or lack of function on, one process. Like, so some of my work is on, well, actually almost all of my work is on a cell signaling pathway, right? And it's throughout the worm, and this particular gene that I work on is expressed almost everywhere. But I want to look at what it does in the intestine. Now, the chances of getting a knockout that is specific to the intestine is vanishingly small, right? So it's, you know, you just wouldn't even bother trying to find it, right? Because it would have to be some kind of very, very specific regulatory change. And so I wouldn't be able to do intestine-specific experiments because it's everywhere, right? So those are the kind of two of the downsides, not all of them, but two of the downsides of using knockouts. Now, knockdowns are a non-heritable, well, they are partly heritable, but not for very long, like 1 or 2 generations. Effect on gene expression that does not change the sequence of the gene being affected.
Oh, okay.
So essentially, this is you being able to mess up a worm without permanently messing it up or its offspring. No, I'm just saying worm because that's just what I happen to work on. With those 2 examples, right, let's look at how you can use a knockdown instead. And so, for example, for the gene which has an early and a late phenotype, right, this was something that I didn't specifically work on when I was a postdoc, but I supervised other people who did. And it was a gene that was involved in vulval development, reproductive development, right, which only develops quite late in worms, same as it does in, you know, most organisms. And so it also had a fairly permanent, lethal, not lethal, but a very bad phenotype earlier on in development, right? So it was very hard to actually see what effect it had on late development. And so instead of using the knockout, at least initially, we used RNAi, which is a form of knockdown. And we're able to knock the gene down specifically after that initial early phenotype or process had already finished. So essentially you can use, you know, like normal function to get past like the first bump in the road, and then you knock down gene function, and then you will see the phenotype later on in the later process that it controls. Right, and so that allows you to kind of have more control over when you express the phenotype that you're interested in, to get around issues with shoot, what's the word? I should know this, I'm a geneticist. Pleiotropic effects, right? Where one gene has multiple, does multiple things in different processes. So if you knock out, it'll affect all of those, right? Not just one specific thing that you might be interested in. Now, does that make sense? So you have control over timing, essentially, of the effect.
Yes, that makes sense.
Yeah, and you can also have control over location of the effect. To some extent, it's not quite, it's depending on how you do it. But you can basically get that RNAi, that knockdown. You can do this in mice as well, and Drosophila, for it to only occur in a particular tissue. So let's say you had you know, a signal produced somewhere in the worm, and then you thought that it was being received in the intestine, right? And so if you knock down the receptor only in the intestine, right, there's nothing for that signal to receive, and then you should see a particular phenotype based on your hypothesis. If you don't see that, then that means there's something else going on that isn't just in the intestine, and you expand your knowledge of the process. Yeah, you can do all kinds of funky stuff. You really can. It's pretty neat. It's a very powerful tool. That's why the people who discovered it got a Nobel Prize. Yeah, unfortunately, I haven't discovered anything quite as amazing yet, so I don't think I'll be getting one. But yeah, and also you can, yeah, you can do lots of funky stuff. You can do conditional knockouts as well, but we're not going to go there. Basically, where you can provide an external stimulus like a compound or whatever, which will create a knockout only at that particular time in response to that particular stimulus. But that's kind of another level above what we've been learning. But generally speaking, knockouts are permanent changes, permanent heritable changes in the DNA of a gene. Knockdown is a transient reduction in output of gene expression, typically by getting rid of the mRNA for that gene. Now, the flip side is that RNAi is not 100% penetrant, right? So when you do gene knockdown, you don't necessarily knock down that gene function to the same extent in every individual or every cell in a particular individual. And so you get much more variable results. And so it's in some ways it's less robust than using a knockout. So they both have their own pluses and minuses. Yeah, I could show you reams of data from RNAi. It's one of the things I use almost exclusively.
All right. Thank you. That helped.
Yeah. And also it gets around the issue of exploring function of genes which are essential, right? Which if you were to do a forward genetic screen, like a mutagenesis screen, you're never going to find that. Well, I mean, I guess you kind of can. There some smart, very smart people do find ways around that, but it's really hard to find mutants in essential genes because they get rid of function at least. Because if you do, then that individual typically doesn't reproduce or survive. Pardon me. And so they'll never come out of that screen. So it's just another, what is it, arrow to the quiver of scientific inquiry, I guess one could say. Questions? What about CRISPR-Cas9? Do you all understand that? Oh, look, there's tumbleweed going by. That's a pretty cool technique. So if you don't fully get how that works, study up on it. Send me emails if you have any questions.
So I guess I have a quick question about the CRISPR-Cas.
Good.
So it chooses either the non-homologous or the homologous recombination. It's one or the other, depending on the circumstances of the test or the experiment.
It depends on whether or not you're providing any donor template DNA.
Okay.
So if you just cut the DNA, like break it apart with a double-stranded break,
Mm-hmm.
it will get fixed by non-homologous end joining.
Okay.
Right. The only way that you can have homologous recombination is if you have something else to recombine with. Essentially, to use a template. It's like a, I don't know. I mean, the kind of equivalent is if you had a tear in a book, right? And you couldn't figure out which pages went together. I mean, it's easier than that because you can look at it. But if you had another copy of that same page or part of it that overlapped the tear, you would use that to line up the break and then fix it through homologous recombination. And so obviously you can provide a template to fix it so it was broken looks like it wasn't broken in the first place, but that's kind of a lot of effort to do nothing. And so really the purpose of the donor template is if you want to introduce specific DNA or a specific mutation, then you provide that in that donor template.
Okay.
So what you could do is say you wanted to change one base in an exon to change an amino acid from, let's say, glutamic acid to glutamine, right? Go from an acid to a base. Then, or maybe two base, I don't know, whatever, two DNA bases. Then you wouldn't get that through non-homologous end joining, right? Because it's just too random. You've got no idea whether it's going to be a deletion or an insertion or what is going to get added or deleted. So what you could do is you could provide a reasonable sized chunk. Let's say that one exon, a few hundred bases or whatever. I don't know the details because I've never done it myself. You provide that, but via other methods like using PCR or whatever, cloning type approaches, you've changed those two DNA bases for that one codon in that exon, right? So now you provide that donor template, which basically has something wrong with it, but most of it is exactly the same as the target gene. So now when you break that DNA, the DNA repair machinery is going to go, oh, whew, that's a breath of fresh air, that's a relief. We've got a copy that we can use to fix this properly. And so it will then essentially flip in that bit that you've provided, and it's got no way of knowing that it's wrong. It simply goes, okay, does this match? Does this match? That's good. Okay, let's flip it in. And then it'll flip it in, and essentially you'll get exactly the same gene, but now you will have those two very specific DNA changes that will change that amino acid from one thing to another. Or, which is even weirder and wackier to kind of get your head around, let's say you wanted to see where that gene was expressed, right? And this is stuff that a friend of mine down in Houston has done. Well, now you can take a green fluorescent protein, pardon me, like what's produced these fluorescent animals here. And now you can put that so that it inserts exactly before the promoter of that gene. Or maybe it inserts at the very end of the coding region, so it gets incorporated into the protein. So now you have an endogenous reporter. You're not providing it externally through your own manipulations. It's actually physically part of that organism's genome. And you do that by, again, you simply have your bit of DNA of interest, whatever that might be, and then that is flanked by existing bits of the either side of the break that you're going to create, so that when you cause that break through the guide RNA binding and the Cas9 cutting the DNA, then when it's fixed, instead of just flipping in like an exactly matching bit with a couple of changes, now you have this much bigger piece which has the flanking regions which match, right? And those match up to either side of the break, and so in the end you get your large extra piece of information or whatever it is that you want gets inserted into that break. And so it's an extraordinarily powerful way of manipulating the genome of an organism, because you can create whatever changes you want. You're really just limited by where you can direct that guide RNA. And there are some limitations. And I think as well, you know, I don't actually know, but I would presume there'd be a limit in the size of the new bit of DNA that you can put into a break site. But I don't know what those limitations are. So yeah, it's an astonishingly powerful technique. And that's how that scientist in China was able to genetically modify the genome of, I think, at least one baby, one embryo who's since been born, to knock out the CCR5 receptor, which is on T lymphocytes. It's one way in which HIV gets into T lymphocytes and causes AIDS eventually. And so he was able to very specifically change just that gene to knock it out somehow. Obviously with a huge ethical implications, but that's another story.
Alrighty, that clears things out. Thank you.
So, I mean, in your future, maybe even in mine, you will almost certainly see the rise of genetic modifications of people.
Yeah, it's kind of worrisome.
It's extraordinarily worrisome. Yeah, but this is, if not this technique, then maybe something that follows on after it. But certainly this technique is capable of doing that.
I think it's kind of worrisome because we have different ethical standards in different countries. So there are different policies and practices that they do or that we do that the other person doesn't.
Right.
So I don't know. It seems like a big mess.
It is, and there'll be that whole, well, we don't want to be left behind, right? If this, I mean, there is very much an international accord, an agreement on not doing this. And so this Chinese researcher doesn't necessarily represent his country's view on this, but simply he was able to, through, you know, lax regulations and weak institutional oversight to do it. I think he's lost his job and, you know, his livelihood and stuff like that. I mean, it's not much consolation, but there were consequences. And he's certainly a pariah. You know, no one really wants him around. But one of the things that I think is particularly interesting, and if you watch, have you watched Gattaca? It's a particularly interesting kind of sci-fi film about eugenics. Yeah, it's really, really well worth watching. The issue is that we don't actually understand how many traits are controlled. So there's a big argument for, say, sickle cell anemia, let's say, or cystic fibrosis, or maybe even some forms of breast cancer, right? To make these edits, if you as an individual and your wife or your partner have those mutations present, to edit them out in your offspring, right? So that you can have children and not worry about them getting the same disease.
Mm-hmm.
And that's kind of like that's a tricky ethical question. But a lot of things like intelligence, right? Sporting prowess, financial success, the kinds of things that, parents might wish to improve for their offspring
Mm-hmm.
are so poorly understood that how would you go about even changing the genome of an embryo to promote those particular traits?
Mm-hmm.
And
Well,
how would you?
doesn't some of it depend on environment and some of it depends on genetics as
Oh,
well?
exactly. Yeah. I mean, that
So,
further complicates things, without doubt. But even the part that is dependent on genetics is dependent on a lot of different genes, in fact, interacting in lots of ways that we don't understand, and lots of things that we don't even know that what there is there, what genes are involved, for example. And so if you did make those changes, if you, let's say you wanted to have, pardon me, someone that was more financially successful, right? That would become wealthier and more
Mhm.
powerful, or whatever. You might end up creating someone that is more of an arsehole, who's more of a sociopath, essentially, because you're selecting for traits that you think go in one direction, but you're also co-selecting for other traits.
Mhm.
So yeah, it's really, really interesting, and Gattaca is an absolute fabulous film to watch on this subject. I'd strongly recommend it.
I'll keep that in mind.
Yeah, because it's very, and it also gets into the whole genes versus environment, and also the matter of probability.
Hmm.
So even if you have a 95% probability of having a heart attack by the age of 50, doesn't mean you're going to.
Yeah, it all depends on the conditions and everything.
Depends on just chance. You could be the 1 in 20 individuals that it doesn't happen to. There's no way of knowing.
Yeah.
So yeah, it's bioethics is a very interesting topic, and genetics does play into that, an awful lot. And it's, there's always this idea of scientists as being, ha ha ha, this is fabulous. We're just going to work on this crazy shit, and we don't care what the consequences are. But scientists do, to a greater and lesser extent, obviously. I mean, we're just people like any others. But yeah, we're not all just mad scientists trying to create superhuman beings and stuff. Not yet, anyway. I'm trying to create superhuman worms. Questions. Come on, keep me fed, ladies and gents. Otherwise, you'll start seeing what kind of news I read.
I had a question, since you're already on that PowerPoint, on slide 8 of the transgenetics. And it's
Transgenics?
showing, yeah.
Oh, okay.
What is it necessarily showing here?
So this is essentially showing the concept of a knock-in, right? So this applies to all kinds of different approaches. It's just a very generic kind of view, but it also describes pretty well what I was talking about with CRISPR.
Mm-hmm.
So essentially what it's saying here is that for any homologous recombination to work, you need to have bits of your template, right, which is the bit at the top, that match something either side of your target.
Oh, okay.
Right, and so essentially what is the case here is that here are these sites of recombination, and this applies as well to CRISPR-Cas9, right? So that homologous recombination versus non-homologous end joining repair, right? This is the homologous recombination approach. And so essentially what the difference between these two things is in the how you have designed the template, right? The bit at the top.
Mm-hmm.
So they both share those matching flanking regions, which allow homologous recombination to a specific site. But this one essentially creates an insert into the gene so that that gene is then disrupted or maybe even modified, right? One way you can think about this is what I was talking about with the GFP reporter, right? So if this is a functional gene and you want to add a GFP reporter protein to it, a green fluorescent protein, and you shine blue light on them and they glow green wherever that gene is expressed. This would be how you would do that, right? But you might put that, maybe you want to actually interrupt that gene, stop it working, and at the same time have a fluorescent marker to tell you that you've done it. Maybe that's one way of doing it. Maybe you don't want to interrupt the gene, but you want to put that marker in frame, right? So it's part of the translated protein. This would also be how you'd do that, right? So this is essentially describing how you would insert DNA into a gene for whatever purpose that you might want, perhaps to disrupt that gene so it doesn't work anymore, perhaps to tag that gene with a fluorescent protein. Maybe even you want to disrupt it with a fluorescent protein, because then you can physically look at the organisms and go, yep, that one worked, because it will be glowing green, and if it didn't do this insertion, then the organism wouldn't be glowing green. There are ways of using that as a tool as well. On the right hand side, it's really a replacement. So now let's say this this particular gene, you could either be something completely different, or this could be a copy of that gene which has been changed in ways that you want to do to see what effect they have. And so now you get a complete flip out. Rather than getting an insertion in to that particular gene, now you just swap out essentially what was there with whatever you want to put in its place. And you get to choose what that is, because you're the scientist designing that particular vector. Yeah, so that's really all that it's showing. It's just different approaches to the same end. So if you look at it down here, essentially that little red bit, that would be potentially
one of those.
Maybe it's this, right? Because you want to insert that bit of donor DNA in, or maybe it's this because you actually, instead of inserting that donor DNA, you want to swap out what was there with that piece of inserted donor DNA. So it's really just different approaches to obtain different ends, really.
Okay. Thank you. That helped.
Cool beans. And there's all kinds of funky stuff that people are doing with CRISPR-Cas9. Crazy stuff. I don't, I struggle to keep up with, to be honest. You can actually do 2 guide RNAs. So one of the risks with CRISPR-Cas9 is off-target effects. So you want to modify just your bit of DNA that you're interested in. You don't want it to create modifications elsewhere, right? Because they may change other genes, or essentially have what's called off-target effects, right? So what you can do now is, if you use 2 guide RNAs, and so you create 2 cuts, or maybe you cut one strand and the other strand using those 2 guide RNAs, that increases the specificity of this process. So it's much more accurate and much less likely to cause changes elsewhere in the genome that you didn't intend. That's actually one of the big arguments against using this to modify humans, other than obvious ethical arguments, is that we don't actually know whether we're causing changes elsewhere. You know, and if that's mice, it's like, oh, sorry, mouse, gets thrown in the freezer. If it's a human, you're kind of stuck with that problem, right? You can't really change it after the fact. Obviously, we don't just go around randomly freezing mice just because we want to, but you can see the differences between mice and humans in terms of experimental ethics. Questions. Go on, ladies and gents. This is it before the exam on Thursday. Rack your brains.
I had another question on PowerPoint 18 about the RNA silencing.
Uh-huh.
And I was just wanting to check my understanding so that the small interfering RNAs are the ones that form the double strand RNA, and they typically are produced elsewhere, and then they typically target the 3' UTRs. Is that typically what happens? Or
that's an interesting question. I've never entirely been sure what the difference is between the various different small letters, then RNAs after, because there are a huge number of them, of which these are just 2 of them. I don't think it's really, if it is split like that, it's not that way round.
okay.
Because I know of microRNAs in C. elegans in worms, whose role is to interfere with translation, right, through the process on the left. No, on the right hand side. Here we go. But I don't know anywhere near enough about it to say if that's the case for all microRNAs.
Oh, okay.
And if small interfering RNAs are solely involved in the degradation of mRNA. I'll look that up and I'll send you an email to clarify if it is possible to be clarified. The real difference between the two, so this really just describes the structure essentially of the RNA, and so both of these are double-stranded. It's just they're double-stranded through the result of different mechanisms, right? This is bidirectional transcription. This is a stem loop formation, right, with an inverted repeat. But they get processed pretty much exactly the same. The stem loop gets the loop chopped off, right? This one gets chopped up into smaller pieces. And then once that goes into the RISC complex, one of those strands gets destroyed, right? So the mechanism by which they're processed is by and large the same. I don't think there are any big particular differences between them. What the starting material is obviously different, but I don't think that necessarily has a huge say in their function. Again, I'll double check. But they're processed largely the same way, but the real big difference, sorry, I've got a rubbish truck going past outside. They're bloody noisy things. The big difference is the degree of matching. So if you have a perfect match across that, the number of nucleotides changes from one type to another. The ones I work with, RNAi, which is generally kind of the equivalent of small interfering RNAs, it's 22 nucleotides. And so if you have a perfect match between that 22 nucleotides and the target mRNA, then RISC will cut that mRNA. If you have fewer matches, perfect matches, and I can't remember there are thresholds, right? This is like dim and distant past. I can't keep all this information in my head, unfortunately. I can't remember the thresholds as to how close you have to get for degradation versus inhibition of translation. But I think it's something like 5 if you have 5 or more mismatches, something like that, then instead of cutting the mRNA, it just sits there. And by doing so, it prevents the mRNA from curling round and the 3' poly-A binding proteins from interacting with the ribosome. And so by doing so, it inhibits translation. So effectively, you get the same result. Pardon me. Presumably, this inhibition of translation is more titratable, right? Because you can just have one risk complex just going all the way around, like a badger kind of cutting up mRNA. Whereas this one, you're going to require one guide RNA and one risk complex per mRNA template, right? To be able to block translation. So maybe it's a little bit more finer tuning in that way. But that's really the big difference between different types of silencing, RNA-induced silencing. One is silencing via degradation of mRNA. The other one is silencing via inhibition of translation, which is effectively the same thing, right? Because you don't care about the RNA, you care about the protein. That help?
Yes, it did. Thank you.
Yeah, but I will look up, because I know for sure that some microRNAs are involved. These are ones involved in heterochronic development in C. elegans. They're involved in binding to the 3' UTR of target genes, and they inhibit translation. But I don't know if that's true for all microRNAs. And obviously, I know that small interferon RNAs are involved in RNA degradation, but again, I don't know if that's true for all siRNAs. I'll have to double check. And usually when I start looking into this, kind of my eyes glaze over because there are so many different types. There's Peewee interacting RNAs. I don't know what they do. That's always one that sticks in my head. Yeah. Anyway. And there are plenty of others as well. There's a whole bunch of them. Any questions? Questions? Speak now or forever hold your peace. It's typically what they say at weddings, but it works here as well. Okay, so what I'll do is I'll look into that siRNA versus microRNA thing for you, Rose. And I'll also get the exam ready and send out that announcement by this afternoon. In the meantime, study the learning goals, go through those, make sure you got all those down. And yeah, just study hard. Particularly my hope is that already the overall exam part that you'll be doing on Thursday, you should all be pretty competent at, right? Because you've been doing attending all these lectures and studying and asking questions and things like that. So hopefully that should be fairly straightforward for you. But study hard the material we've done recently, because that's really quite tricky. It's quite difficult to get your head around and keep things separated, particularly between like inducible and repressible gene expression systems, the two different systems in bacteria, and then all of the different varying levels of craziness that go on with eukaryotes. If you have any questions, just send me emails and I'll answer them within a day.
So just to be sure, the exam 4, I think, it's over the prokaryotic and eukaryotic gene regulation and transgenics. It's just those 3.
Yeah, that is what it is.
Okay.
Not a trivial amount of
Alrighty.
material, to be honest.
Well, it's very, very heavy on the lectures.
Mhm.
It's just not like 4 or 5 of them.
No, no, but the concepts involved are pretty weighty. And obviously we spent a bunch of time on the LAC and the trip operons, so those were guaranteed to be in the exam in some form or other. And we've also spent a whole bunch of time on control of gene expression in eukaryotes. So, you know, chromatin, RNA processing, control of transcription, all of that kind of jazz obviously are going to be on the exam as well.
Alrighty.
Cool.
And you did say that we had all day on Thursday to do it?
Yes, it will shut at midnight. You will not be able to take it after midnight. And
Okay.
so presumably one would want to start it before 11 o'clock, essentially.
Yeah.
But it will be available after at the start of the normal class period, which is technically 9:30.
Okay.
Right, so the exam will pop up. You won't see it before then because it's only made available at that precise time. It'll be from 9:30 until midnight, essentially. And also you're going to have the 2 parts, so you don't have to do them both, you know, in order. You can do one in the morning and one in the afternoon, let's say. There's a lot more flexibility about that. But I will be available in the morning during class period, at least for half an hour. You know, if no one shows up, I'm not gonna stay hanging around, just in case any questions pop up. And if they do, then I can make changes both within that, and then I can send around an announcement and let everybody else know that hasn't taken it yet. So it's really up to you when you do it. If it suits you better not to do it during normal class times, then do it outside of that time. If you're worried that there's going to be stuff in there that you don't fully understand and you want to ask me a question, then I'll guarantee that I'll be around at least for the first half hour, 40 minutes of the exam to answer those questions. So it's really whatever works best for you.
Alrighty,
Yeah, because obviously after that period, you know, I've got other stuff that I might be doing. I might go taking the dog for a walk or whatever while you're taking your exam. And so I won't be able to answer any questions you have there and then. So I mean, there's pluses and minuses to both approaches, but it's really up to you.
Is there a time limit to the exam?
Yes, there will be time limits. Each exam will be whatever half of the class period is, which is 75. It'll be like probably 40 minutes, 35 minutes, something like that. For each exam, each will be 20 questions.
Oh, that's not bad.
Yeah, so the total number of questions, the total amount of time will be the same for exam 3, but it will be split in half.
Okay.
But yeah, I'll address all those details in the announcement when I send it around. And if I forget any, remind me, because you know I haven't got a brilliant memory.
Sounds like a plan.
Cool beans. You all right, Rose?
Yes, I'm fine. I'm just listening because everyone's asking already the questions that I have, so it's fine.
Good. All right, and if you have any more, just send me emails, and I'll get back to you with ideally within a few hours. I'm going to be on my computer the whole day doing both the exam and other things that I've got to get done.
Thank you, Professor.
You're very welcome. Thank you all.
Alrighty, have a good day.
You too.